Good morning, everybody. Would like to start. Maybe if you can come a little bit closer, that will be better, and we'll give the new people who arrived to sit on the back. Dear Dan David laureate, dear faculty member, dear student and dear guest, and ladies and gentlemen, it's really an honor and privilege to open this special symposium on memory and mind, biological and digital, part of the Dan David Prize. The Dan David Prize, as you know, is already given for the 13 years. It's been becoming a world-known and prestigious prize, and usually the prize is composed of 3 dimensions: past, present, and future. And luckily the theme of the present prize was memory and mind, and this is exactly the daily life of the Sagol School of Neuroscience, which deal with neuroscience and brain, and investigate brain function and dysfunction. And we are happy to have here today in the present dimension, which is on the title of Combating Memory Loss, Professor Brenda Milner, Professor John Hardy, and Professor Peter St. George-Hyslop. Thank you for coming. In the future dimension, which was titled Artificial Intelligence, we have Professor Mavri Minsky. And I think that the nice thing is that these 2 dimensions interact with each other. There is memory formation, memory loss, and then artificial intelligence that might help us maybe to create new memories. And on this frame, we have decided the School of Computer Science and the Sagol School of Neuroscience to have this joint symposium that will deal with these topics from different directions, from the direction of memory formation, memory loss, and artificial intelligence. And this is part of an ongoing activity that the School of Computer Science and the School of Sagol Science has, and it's been reflected in the last year new program for bachelor degree that we opened, which is Psychology and Computer Science, which gives the student the ability to have both information from the world of psychology, biology, and computer science, and to investigate the brain from different directions. And this is part of the idea of the Sagol School of Neuroscience, which is bringing researchers from different directions. We have 100 researchers coming from 7 different faculties, including computer science. And this is, I think, the idea of bringing researchers to investigate the brain from different directions. We're going to have a very busy day. We'll have 4 lectures by the laureate and few lectures by faculties of Tel Aviv universities. And during lunch, we'll have poster presented by students in the foyer. And a part of these posters, a special section, will be posters of students that are supported by Teva company. And Teva initiated at lunches last year a very powerful program, which is called NNE, National Network of Excellency. And the idea is to support research in brain science. And we are very happy that Tel Aviv University won many of the prizes of the fellowships, and the students are going to present their work during lunch. So without further ado, I would like to invite Professor Yoav Henis, the Vice President for Research and Development of Tel Aviv University, to greet you. Please.
Dan David Prize laureates, Professor Hardy, Professor St. George-Hyslop, Professor Brenda Miller, Professor Marvin Minsky, dear Tel Aviv researchers, students and guests. I'm honored to open the Dan David Symposium on Mind and Memory, Biological and Digital. Our brain is made up of millions upon millions of neurons which communicate in complex ways which are still a mysterious unknown. Understanding the brain and the integration of its activity into what one may call mind is an ultimate challenge which is at the frontiers of modern biology and medicine. It is important not only for understanding how this most complex of organs functions, but also for diagnosis and development of novel treatments of many devastating neurodegenerative diseases which have multiple effects on modern society. These important aspects were clearly evident in the Dan David Prize this year, in which 4 laureates actually deal with various aspects of brain and mind research. I truly hope that the current emphasis on brain research will lead to novel understandings of brain and mind and to future development of therapeutic approaches. I wish you all a wonderful and interesting symposium and a lot of success in your future research for the benefit of science and society. Thank you.
Thank you very much, Professor Henis. And I would like to call the co-chair of the session, Professor Nachum Dersovich from the Computer Science School, to introduce, to welcome the ambassador. Please.
Good morning. It's a sublime honor to call upon His Excellency Mr. Matthew Gould, the British Ambassador to Israel, to greet this symposium. Mr. Gould has been British Ambassador to Israel since September 2010. Over these years, he set up the UK-Israel Life Sciences Council and also the UK-Israel Tech Hub, as well as the £10 million Britain-Israel Research and Academic Exchange Partnership in regenerative medicine. Earlier in his career, Mr. Gould served as Private Secretary for Foreign Affairs to Prime Ministers Tony Blair and Gordon Brown, and as Chief of Staff to 2 Foreign Secretaries. His diplomatic postings include Washington as Foreign Policy Counselor and Representative of the Joint Intelligence Committee, Iran as Deputy Head of Mission and as Acting Ambassador, Pakistan just after 9/11, heading up the embassy's political section, and Manila as the political officer, where he was awarded the Most Excellent Order of the British Empire for having set up a program of cooperation between British and Philippine police and detectives for tackling child abuse. He also established a unit of the Foreign and Commonwealth Office to help combat forced marriages around the world and aid its victims. Mr. Gould speaks some Farsi and some Hebrew, though I suspect he'll speak here in English. He read philosophy and theology at Peterhouse College, Cambridge. Before that, he spent a year teaching English and mathematics at a secondary school in the tribal trust lands in northern Zimbabwe. He has published a scientific paper in the journal Sociobiology entitled The Frequency of Termite Isoptera Damage to Tree Species in Namakutwa Forest, Tanzania, on termite feeding preferences in the dry coastal forests of Tanzania. Not only is Mr. Gould a longtime patron of the sciences and of British-Israeli cooperation in the sciences, and a scientist in his own right, but furthermore, all 3 present-time laureates are British, making his gracious presence at this symposium particularly apt. Mr. Ambassador.
Thank you very much, Nachum. You've done me an enormous honor, I have to say, most of all by lying to the audience and saying I'm a scientist. It's incredibly kind of you, and actually one of the nicest things that anyone said about me since I've got to Israel, and I will treasure that. It's a real pleasure to be with you today for both personal reasons and professional reasons. I will come on to the professional reasons, but let me start with the personal, and that is I stand in the very one of the reasons that it is so nice to be speaking to an audience of doctors and people active in the field of medical research is I place myself very firmly in the Jewish tradition of hypochondria. Indeed, this very morning before coming here, I went to see my doctor, And on a number of occasions, I have diagnosed myself with early onset Alzheimer's. And so it'll be a great pleasure for me to be able to introduce our experts in the field. It does all those, though, remind me of my favorite Jewish joke. And having an audience like this is too good an opportunity to miss telling it. And it's this. There is an Englishman, a Frenchman, and a Jew lost in the desert. And they're crawling along. They're exhausted, and the Englishman says to himself, "I'm hot, I'm tired, I must have a pint of beer." And the Frenchman is crawling along, and he says, "I'm hot, I'm thirsty, I must have a cognac." And the Jew is crawling along, and he says, "I'm hot, I'm thirsty, I must have diabetes." The honor of speaking to you today is afforded to me by the presence of not one, but was, as mentioned, three of the Dan David laureates. And it's for me a sort of fantastic honor for the UK that so many of our scientists should be honored in this way. Just to run through them, Professor Hardy is the most cited Alzheimer's researcher in the UK, the fifth most cited internationally, was the first to discover a mutation in the amyloid gene, which plays a key role in the neurodegeneration associated with Alzheimer's. We have Professor St. George-Hyslop, who by discovering two genes responsible for early onset Alzheimer's has made early diagnosis and treatment of a disease possible, often before brain damage occurs. And then we have Professor Milner, whose work has led to many landmark discoveries in the study of human memory and the brain's temporal lobes. And Professor Milner obviously is here from McGill University, but as she very proudly showed me her British passport, I am very proudly going to claim her as a British recipient, and the Canadian ambassador isn't here to tell me I can't. The Dan David Prize is, as was said, divided into three categories, and this year all three are devoted to the common theme of memory and what many now regard as the final frontier of science, namely the mind. The category of the present, devoted to researchers combating memory loss and neurodegenerative diseases, is particularly relevant to the UK because this quiet crisis of memory loss, dementia, neurodegeneration is perhaps one of the most pressing socio-medical issues of our day, and has been made by our Prime Minister David Cameron a very personal priority. And he called a summit on the issue at 10 Downing Street not very long ago that one of the laureates today was present at. And we estimate the global cost of managing dementia exceeds $600 billion a year. But more than the cost to the world's economies is the cost to hundreds of thousands of families who have the extraordinary pain of losing loved ones slowly to dementia. And that is a huge personal burden. And we believe that Britain and Israel are very well placed to collaborate on this issue. Both countries excel in neuroscience. Both countries are the home of pioneering work in the field, new drugs, innovative ways to tackle the challenge of dementia. And it's a source of real pleasure and pride to us that Teva has now committed to a 3-year dementia research program, providing funding of $1 million for early stage work in the UK. And it's also a source of real pride to us that the British Neuroscience Association is teaming up with the Israeli Society for Neuroscience and announced a joint symposium on dementia, which will be held later this year in Eilat. And all this is coming at a time when the relationship between the UK and Israel in science has never been stronger. And, I mean, it was very clear to me when I became ambassador that the potential for this relationship is huge. And we are both, as my Foreign Secretary and boss put it, scientific superpowers. We both have disproportionate numbers of Nobel Prize winners, disproportionate numbers of world-class universities and world-class laboratories. And there is so much more than we can do, that we can do together than we can separately, that it seems to us a crying shame that we're only just starting to fulfill the potential of this relationship. And for me, as ambassador, there's no better model for how I want the relationship to be between the countries than the model of collaboration between our scientists, free from politics and focused on making progress for the benefit of all humanity. That, for me, is a very pure and rather wonderful model. And so it's no coincidence that when my boss, the Foreign Secretary, William Hague, came here last, the very first thing he did was to go to Hebrew University and sit down with the Israeli science minister and sign a memorandum between Britain and Israel to promote scientific collaboration. And that memorandum set out 4 priority areas for our research, of which one is neuroscience. And it's also no coincidence that when his boss, the Prime Minister, David Cameron, was here just a few weeks ago, he, with President Shimon Peres, did an event to promote our collaboration in science and met a series of Israeli scientists doing very exciting work with the UK. And so it's a real pleasure for me as ambassador to be invited to help open this symposium and honour the 3 British recipients of a Dan David Prize. And I just wanted to leave you with one thought, or rather 3 thoughts, that have struck me about this symposium, because I think there's something very special about what you're doing today. The first thing that I thought was special was its extraordinarily international aspect. This meeting of minds to discuss the mind brings together scientists from all across the globe, not just Britain and Israel. And I think you are broadening more than just scientific horizons today. And I think this is a wonderful example of the power of science and scientists to reach across international borders. The second thing, which I think is very special, is the interdisciplinary nature of what you're doing. And looking at the lineup for the speakers today, it's very striking how your discussions are pulling in people not just from the faculties of medicine, but from computer science and nanoscience and biological science and psychology. And I think this is something very important, because the ability to transcend the traditional barriers and silos of scientific disciplines and reach beyond into other areas of expertise requires real dexterity, but the rewards for doing so are huge. And as the Jewish saying goes, who is wise? He who learns from every man. And then the third thing, the last thing that I'd say about what you're doing today, is just to point out the cooperative nature of this symposium. It's quite rare in my experience to have a symposium like this, which makes so much effort to make itself open and accessible, not just to the scientists who are taking part, but to members of the public as well. And so the overarching message that I'm left with is that this symposium shows that science is for everybody. Not only that it affects everyone, but that contributions can come from everywhere. So I wish you all a very successful and fruitful symposium.
Thank you, Mr. Ambassador, for the great words and description of this event. And I would like to call Dr. Danny Frankel to introduce the first speaker.
Distinguished guest, audience, I'm very happy and honored to present Professor John Hardy. Professor John Hardy is the head of the Department of Molecular Neuroscience and chair of Molecular Biology of Neurological Disease at University College London Institute of Neurology. Professor Hardy was the first to discover a mutation amyloid gene encoded the amyloid precursor protein, which plays a key role in neurodegeneration associated with Alzheimer's disease. His research interests are in the genetic analysis of the disease. Historically, he was working on the genetic analysis of Alzheimer's disease and other dementia. More recently, he focused on Parkinson's disease and other movement disorder and motor neurons. His research aimed to develop an understanding and the underlying genetic of the disorders, both in understanding disease mechanism and to help the search for treatments. With over 23,000 citations, Professor Hardy is one of the most cited Alzheimer's disease researchers in the world. In recognition for his exceptional contribution to science, he was elected as a member of the Academy of Medical Science and of the Royal Society of London. Professor Hardy received numerous distinguished awards and honors, among them the Potamkin Prize for American Academy of Neurology for Alzheimer's Research, the Khalid Iqbal Award of Lifetime Achievement in Alzheimer's Disease Research, and the IFRAD Prize for Alzheimer's Research. Professor Hardy, I'm very honored to invite you to give a talk.
It's a real pleasure to be here today. I've had a great trip. My wife and I have had a great trip here to Israel. We've seen great hospitality and, been around the country. It's a beautiful place to come to. I've really enjoyed it, and I'm looking forward to the Israeli-British Neuroscience meeting later in the year. So I'm going to talk about whole genome analysis of Alzheimer's disease, and really I think I've got a couple of messages really. The first is that through this we're beginning to trace out the pathways to disease. The second is really to show you how genetic analysis has become so much more powerful over the last 5 or 10 years and now allows us to really approach any disease area in a systematic fashion and find the genetic risk. So what I'm saying, although of course I'm going to talk about Alzheimer's disease, is applicable to other neurodegenerative and neurological diseases, but also applicable to diseases across the spectrum. This is how we think these days about genetics. This is, if you like, a graph of how we think of genetic risk. On the left-hand side at the top here in blue, if I could, yeah, it's a bit difficult for me to see, but here, these are what we think of as genetic diseases, where they're rare in the population. This is frequency along this axis. But if you have these variants, you will get disease. This is the risk here. We've been able to find this type of genetic risk for 20 years in a systematic fashion by the process of positional cloning, which I'll show you in a second. So this type of genetic risk we've been able to find for a very long time, a very, very long time. But as I'll describe, we can now find it much quicker than we were able to in the past. Down here in the bottom right, this is normal genetic variability. There's about 200 people in this room. This is variability that 40 or 50 of us in this room will have, but it only influences our risk of disease by a relatively small amount, between 1.1 and 2-fold. So these are small effects on risk that are present in the population. And we've been able to find these, as I'll show you, in a systematic fashion for about the last 5 or 6 years by a process called genome-wide association studies. In these studies, we test genetic variability across the genome for association with disease. We do essentially 500,000 chi-squared tests for genetic variants across the stepped across the genome for risk to disease. So we can find this type of variability systematically as well. In other words, we can, if it's there, we can find it. Finally, in the middle here, this has been the most difficult area, and this is just about soluble as well now. This is variability that perhaps one or two of us in this room will have, so it's rare. But it doesn't cause disease, but it massively increases your risk of disease. So this is so rare that we can't really find it through association studies, and yet it does not lead to extended families with disease, because I might have the gene and the disease, but my father had the gene but didn't get sick. His mother had the gene and did not get sick, but her sister might have had the gene and got sick. In other words, it doesn't inevitably lead to disease, it just increases your risk. This type of variability we can now find through sequencing a whole exome, that's all the protein coding parts of the genome, or whole genome sequencing. So every part of this curve we can now find the risk in a systematic fashion. The other point that I'm going to draw out as we discuss the genes is that whatever the type of risk these genes encode, what we're beginning to find is that all the genes for particular diseases map to relatively few pathways. In other words, as we find the genes for disease, what we're really doing is identifying the biochemical pathways which are important in the pathogenesis of the disease. This is the pathology of Alzheimer's disease. Here you see the plaques made up of the amyloid peptide, and then these stringy bits are tau filaments, phosphorylated tau. So this is the pathology of Alzheimer's disease, which underlies the clinical features of the disease. And really, I think that modern work on Alzheimer's disease started with a pair of papers from Glenner and Wong in 1984. In the first paper, Glenner isolated the amyloid from a case of Alzheimer's disease. And in the second paper, and this is the second paper, in the second paper he took a brain of somebody with Down syndrome. People with Down syndrome have long been known to get Alzheimer pathology when they reach their 40s and 50s, and got the sequence of the amyloid peptide from there as well, and noted it was the same, and wrote the abstract, which I'll read. The amyloid protein from a case of adult Down syndrome was isolated and purified. Amino acid sequence analysis showed it to be homologous to that of the amyloid peptide of Alzheimer's disease. This is the first chemical evidence of a relationship between Down syndrome and Alzheimer's disease. It suggests that Down syndrome may be a predictable model for Alzheimer's disease. Assuming the amyloid peptide is a human gene, it suggests that the genetic defect in Alzheimer's disease is on chromosome 21. All of that is essentially true, and implicit in what Glenner said is that he clearly thought that overproduction of amyloid was one route to getting Alzheimer's disease, because of course people with Down syndrome have got 3 copies of chromosome 21, whereas the rest of us have got 2 copies, and he is implicitly suggesting that people with Down syndrome simply get Alzheimer's disease because they make too much amyloid. And that is really the basis of the amyloid hypothesis of the disease. This is a very old slide now. This is from the paper that was mentioned, Finding Amyloid Mutations, and I show it because it explains how we in general find genes which cause Mendelian autosomal dominant disease. And what you're looking at here is a grandfather with Alzheimer's disease. If I can move. Grandfather with Alzheimer's disease, 2 sons with Alzheimer's disease, both dead. These 2 sons married 2 sisters, and these, okay, I'll use a pointer here. These These 5 people with Alzheimer's disease inherited the top part of the chromosome, as did their cousin here. These all got sick. But crucially, these 2 individuals here, this individual did not get sick and got the top bit of the chromosome. This individual here did not get sick and got the bottom bit of the chromosome. So the simplest explanation is the disease gene must be between here and here. There are about 30 genes in this interval, but one of them is the amyloid gene. And when we sequenced the amyloid gene in this family, we found that these individuals had an amino acid change. So this was the work that was referred to. This is the amyloid mutation causing disease. Now, although this is a very simple slide, what this actually shows is 30 person years of work. This took a group of 6 of us 5 years to do. So this is 30 person years of work. Now, this same experiment would probably take a competent postdoc about between 1 and 3 months to do, because of the developments in genetic technologies to do with, well, of course, knowing the human genome sequence and therefore knowing the position of every gene. PCR was not invented at this time. That, of course, helps enormously. The whole genome arrays allow us to assess all the markers in parallel in the genome, so we could do this 2 orders of magnitude more quickly. So this was an amyloid mutation causing Alzheimer's disease. That was an amino acid change. The French group in 2006 reported this type of family, which we have now identified about 8 or 10 versions of in the UK, with amyloid gene duplications. And what you're looking at here is a gene chip analysis of chromosome 21 here. And if you can see here, what you're scoring is alleles on this chromosome, AA, AB, BB. The chromosome is lying along here, but you can see in this part of the chromosome we've got an extra allele. That's because there's a gene duplication here, and the amyloid gene is within that duplication. And this really finishes Glenner's work, in a sense, because this really proves that Glenner was right when he said that people with Down syndrome get Alzheimer's disease because they have an extra copy of chromosome 21. In these individuals, they don't have Down syndrome, but they do have the extra copy of chromosome 21, and sure enough they get Alzheimer's disease when they're in their 50s. Another point I want to make then at this slide is that when you find a locus for disease, you find many different variants at that locus which influence risk. So this is one example where you can have amino acid change or gene duplications. I'll come on to other examples later on. So this work allowed us to formally state the amyloid hypothesis. As I say, it was implicit in what Glenner had said in the 80s. Now, when we found amyloid mutations, we had about 30 families with early onset Alzheimer's disease. The family I showed you had an onset in the 50s. That meant that most of our families did not have amyloid mutations, so there must be other genes, and that the race to find the other genes was run by Peter Hyslop, who's going to talk about that, I'm sure, in a few minutes. And this is, the structure of the gene he found, presenilin 1 here, and this is the amyloid precursor protein in the active site of presenilin 1. Every residue in red is a residue at which a mutation has been found which causes Alzheimer's disease. And when Peter found presenilin 1, we did not know what its function was. We now know that its function, one of its functions, is to cut the amyloid precursor protein here. These are 2 aspartic acid residues. They act like a pair of scissors, and they cut the amyloid precursor protein here. So their function then is to be part of the metabolism of the amyloid precursor protein. And that harks back to what I said a minute ago at the beginning, that when you find different genes for disease, what you're doing is delineating a pathway. And sure enough then, the initiation of this pathway seems to be the cleavage of the amyloid precursor protein. Families with amyloid mutations in general have an onset age in their about 55. Families with presenilin mutations tend to have an onset age between about 30 and 50, although there are exceptions to those rules. But they are otherwise typical in terms of their pathology and clinical presentation of typical late onset Alzheimer's disease. What I've been describing so far are mutations which cause Alzheimer's disease. I said, though, that when you find a gene for disease, what you find is at that gene you find multiple alleles which are associated with different amounts of risk. And related to that statement was this paper, very interesting paper from the Decode group, which is a mutation in the amyloid gene which protects against Alzheimer's disease. In other words, the mutation I showed you before in the amyloid gene is one which causes disease. This is a mutation which protects against Alzheimer's disease, and the reason it protects against Alzheimer's disease is because it inhibits the metabolism of the amyloid precursor protein. And this is important from both a theoretical point of view, but also from a treatment point of view, because this suggests that if you have a lifetime inhibition of the metabolism of the amyloid precursor protein, you have a lower risk of getting Alzheimer's disease. And so this really does point the drug companies at the idea that they should try and reduce the production of amyloid in the brain as a route towards developing therapies for disease. All the families with clear autosomal dominant Alzheimer's disease have mutations in the amyloid precursor protein or the presenilin genes. All of them. So there are probably no other genes which have a one-to-one relationship with the disease. What I'm going to talk about now is the risk variants, the variants that many of us in this room will have which predispose to disease. I show these slides because they tell a story about how these studies are organized. These are from 2009, and what they describe is huge collaborations where groups from all over the world, this is UK-led, this is a French-led, and this is a US-led study, with each of them have got hundreds, well, not hundreds, but 70 to 100 authors because they involve all the groups collaborating and putting thousands of cases and thousands of controls together and then testing for association across the genome. And these are the first of these studies, which were 2009, and they are beginning to yield, as you can see, consistent results. So this one found clusterin and PCALM, and this one found clusterin and complement receptor 1. And as we've gone on now, we're finding more and more genes here. Now, this is the most recent study which came out earlier this year, and you can see that it involves 74,000, the analysis of 74,000 individuals. And as we increase the size of these studies, we find more and more loci involved in disease, and this is that analysis here. And what you have here is the chromosomes laid out in order along this axis, and then the p-value, the statistical significance here. You're doing about 500,000 chi-squared test because you're testing so many polymorphisms, so you have to set a Bonferroni correction about here, and everything which exceeds that exceeds that point here is declared as a gene for a locus for disease. ApoE is easily the most important, and I'm sure that Danny Michaelson will talk about this. It is easily the most important. It has an odds ratio of for ApoE4 of close to 4, and actually this is truncated. It would go way through the ceiling here. These other genes all have odds ratios between 1.1 and 1.5. Essentially, as we increase the number of samples we analyze, we will continue to increase the number of loci we find. It's a bit boring, though, just to have a list of genes involved in disease. That's more of a catalogue than an insight. But it is clear that we are, when we look at the functions of these genes, we are gaining insights into the biology underlying the disease. And this just looks at the first 8 loci. What I'm saying is true of all the loci whose function we know. And I'll list them here. ApoE, as I mentioned, we know that that's involved in cholesterol metabolism. Clusterin, we also know, the other name for clusterin is ApoJ. We also know that this is involved in cholesterol metabolism. It's part of the complement cascade, the innate immune system. PCALM is involved in endosomal vesicle recycling. We don't exactly know the function of PCALM. I think Peter might talk about this, but we believe it might be indirectly involved in amyloid metabolism. ABCA7 is definitely involved in cholesterol metabolism. Its homologue ABCA1 is the gene in Tangier's disease, which is a cholesterol storage disease. Complement receptor 1 is obviously involved in the complement cascade, the innate immune system. BIN1, like PCALM, is probably involved in endosomal vesicle recycling, and again might indirectly be related to APP metabolism, though this is not sure. And then MS4AE is involved probably in the complement cascade. So we're beginning to see then that, of course, these are broad categories. The innate immune system is a broad category of gene function. Cholesterol metabolism is a broad category of function, and endosomal vesicle recycling is an important and central part of cellular metabolism. But these are comparatively small proportion of genes in the genome. So we are beginning to see then that these processes are what is important in Alzheimer's disease. And the genetics is telling us what parts of those processes are key components of the disease. And that is that point which I've made in a kind of anecdotal way is made in a systematic way by this paper from Leslie Jones. So we're beginning to see pathways to disease. And actually, if somebody declared that they thought that they had found a gene, a locus for Alzheimer's disease, which wasn't in these pathways, I would have a high degree of suspicion that they were not right, because it's turning out to be a very robust finding across groups. So we've dealt with then with these forms of the disease, these genes here, amyloid and the presenilin. We've dealt with these here, these common variants. Now I'm going to talk about the work that's really only just started here in exome sequencing the genome to try and find these high risk variants. And last year we were fortunate, I don't know, this is perhaps, I don't know how easily you can see this, we were fortunate in finding, we and the DECO group were fortunate in finding mutations in a gene called TREM2 involved in Alzheimer's disease. These are variants which are present in about 0.5% in the general population and about 2.5% of Alzheimer cases. We found this, the way that we found this gene and the way that the DECO group found the gene were different. The DECO group looked at the entire Icelandic population and treated the entire Icelandic population essentially as one large family. And looked to see what parts of the genome all the people with Alzheimer's disease shared, which was less often shared by their compatriots. We had came at this in an entirely different way. We had been sequencing rare recessive dementias in the Turkish population. Obviously there's a lot of consanguinity in the Turkish population, and we had found homozygous mutations in this gene called TREM2 in Alzheimer, in an early onset dementia in Turkey. And when we looked at the mutations that we had found in the Turkish disease in a homozygous fashion, we then looked in our English Alzheimer series and found we had got the same mutations heterozygous in our Alzheimer cases in England, so we came at this from an entirely different perspective. Interestingly, so these papers came out last year. This paper from 1983 actually makes the same point as we had made over, well, nearly 30 years later. Ted Bird had identified a recessive disease with this, which is the same disease we had found in Turkey, and had shown that some cases of that syndrome had actually got Alzheimer pathology. And he had suggested that whatever caused this rare recessive dementia was also implicated in Alzheimer's disease. We were not aware of this. You can see, in fact, the disease had changed names, so we didn't find it by a literature search, and the gene for the disease had not been described there. But Tom wrote to me very politely when our New England Journal paper came out and said, "Nice finding, shame you were 30 years late with it." But it was a nice letter, though. So the nice thing about TREM2 is that we actually have a good idea, I emphasize it's an idea, a good idea of its function. It's a microglial gene, and its role seems to be in holding microglia in a phagocytic phenotype. When microglia are exposed to something like amyloid, they first try and engulf the amyloid, that's their phagocytic role, and when that process fails, they switch to an inflammatory process, pump out cytokines to bring other microglia into the vicinity. And TREM2 is one of the receptors which holds the microglia in this phagocytic role. So its role at this point is phagocytosis. And so at this stage in Alzheimer's pathology, there is amyloid in the brain, but that amyloid is not provoking a catastrophic reaction, tangle formation and cell death. What it is doing is that amyloid is being engulfed and dealt with by the microglia. When this fails, then it's switched to cytokine production and to an inflammatory response. And it seems that if you have only one copy of TREM2, it makes it more likely that your microglia will switch from the phagocytic role to the cytokine and inflammatory production role. And so this is likely to be a key part of Alzheimer's pathogenesis. I should say that people have been saying that inflammation, that microglial activation was important in Alzheimer's disease for something like 30 years. What this shows, really what these genetic findings show, is that genetic variability in that process is key in determining who develops disease and who manages to maintain their amyloid deposition without having a catastrophic reaction to it. So this is now where we're up to, echoing that first review slide. We've got all of these genes down here. These are the GWAS hits. ApoE4, easily the largest effect, ApoE4 homozygosity here. Here we have the Mendelian genes here, which I've mentioned, amyloid and presenilin 1 and presenilin 2, and then this is TREM2 here, and we've actually just subsequently found another gene, PLD3, which is also in this area here and probably involved in amyloid production. So we are beginning to fill in this, if you like, the architecture of risk for Alzheimer's disease. We've got a lot more genes, I'm sure, to find, especially in this area here, but I would suspect that those will continue to map to the pathways we have already been discussing. So these are the lessons we can draw from Alzheimer genetics. The Mendelian genes are all involved in amyloid production. There's also protective variants, and these reduce amyloid production. The risk maps to definable pathways, including the innate immune system and the complement system. Some of the genes appear to be amyloid response genes, and I would put TREM2 in that. Endosomal recycling genes may be involved in amyloid production. This is an area of research. It's not absolutely certain yet that that is the case, and I would say that all of the genes are consistent so far with the amyloid hypothesis first expounded by Glenna all those years ago. Thank you very much. Shall I take questions? No. Okay, you can ask questions at the coffee break.
So I'm humbled to introduce Professor Peter St. George-Hyslop. Professor Peter St. George-Hyslop is at the Wellcome Trust Principal Research Fellow at the Department of Clinical Neuroscience, University of Cambridge, and Director of the Tan Center Research in Neurodegenerative Disease in the University of Toronto. He was the first to discover key mutations in proteins involved in the early onset of Alzheimer's disease and implicated in late onset of this disease. Peter St. George-Hyslop has won one of Europe's top health awards for his pioneering work on the roots of neurodegenerative diseases, including the Bial, I hope I'm saying it correctly, a Merit Award in Medical Sciences. Among his honors and awards are the Francis McTone, I hope I'm saying it correctly again, Prize from the Canadian Neurological Society, and an award for medical research from the Metropolitan Life Foundation and Distinguished Scientist. He won several more awards, and he is also a member of the American Society for Clinical Investigation, a fellow of the Royal Society of Canada, and a foreign member to the Institute of Medical of the National Academy of Sciences, and a fellow of the Royal Society of London. Please.
Let me begin by first of all saying what a tremendous honor it is to be awarded the Dan David Prize. I think my colleagues were equally surprised as I was by being told that we'd won this prestigious prize, and it is truly a great honor. Let me now tell you a short story in the next 40 minutes or so about the biochemical genetics of Alzheimer's disease. And in choosing this story amongst all the other stories that I could have told you, I decided on this story not only because it's a nice, long, scientific, technical story that goes all the way from humans down to almost atomic detail, but perhaps more importantly, as you will see, it tells a human story. And I think the human story may in fact be larger than the actual scientific story itself. Specifically, what I'd like to do is to show you how the analysis of an obscure illness in a Russian Jewish family led to the discovery of the presenilin genes, which, as John Hardy told you a few moments ago, is one of the key players in the biology of Alzheimer's disease. I'd like to show you some data to convince you that the presenilin proteins form a membrane-bound protein complex with several other proteins, and that this complex functions as a very unusual enzyme that cleaves membrane proteins within the transmembrane domain of those proteins in the lipophilic environment of the membrane, which is, I think most physical chemists would tell you, is almost a biological impossibility, and yet it does in fact occur. I'd also like to show you that this presenilin-dependent cleavage of these membrane proteins, particularly key substrates like the Notch protein, is essential for life, all the way from very archaic bacteria all the way up to humans. This protein complex is indeed a vital machine. I'd also like to show you that aberrant cleavage of one of the substrates, the amyloid precursor protein, as John Hardy told you a few minutes ago, causes the most aggressive form of Alzheimer's disease. And finally, I'd like to show you the technical details of how structural biological investigations of this presenilin complex has begun to show us how the machine actually works at almost an atomic level to cut these substrate proteins. And this knowledge, with a bit further refinement, should allow us to eventually design very substrate-specific drugs that act to prevent the cleavage of amyloid while still permitting the necessary cleavage of the other substrate proteins, such as Notch. So the story begins in 1844 with the birth of Hannah. We don't know exactly where Hannah was born. It was probably either in Vilnius in Lithuania or Riga in Latvia. What is known is that she spoke the Lithuanian dialect of Yiddish, which means that she and I could not have spoken to each other. In her 20s, by presumably a traditional Jewish marriage, she was married to Shlomo, who was born and lived in Bobrusk, in what is now eastern Ukraine. And initially, they had a happy marriage, and they gave birth to 9 children, some, not all of which, are shown here. But at about the age 40, Hannah began to develop memory problems, and survivors of the family who knew her recorded that at about the age 40, she became forgetful, then she began to have difficulty looking after herself, and many of her children recall that they had to wash her hair, feed her, etc. And the symptoms that they describe are the symptoms that we now see of a patient with classical Alzheimer's disease. However, that wasn't the only affliction that happened to Hannah and her family. There was an attempt, or actually a successful assassination of a Tsar of Russia, and what followed was a hideous series of pogroms. And initially, Hannah's village was not visited by the pogroms until 1891, when one came to her village. The village was essentially destroyed, and in the melee, the fear, the terror, Hannah, obviously unable to understand what was going on, suffered a massive cardiovascular collapse and died. She was buried that night in the garden to prevent desecration of her body, and the family moved away to another village. However, they did not escape the further pogroms that happened again between 1903 and 1905. And these ones were particularly vicious and nasty. And the family decided to have a diaspora and to move away. First one or two of the sons, then several of the daughters, and eventually even Shlomo himself moved away to the United States. But they didn't move as a single entity, and they didn't move to the same place, but instead moved throughout different parts of the United States, where they had a typical immigrant existence, and where things initially seemed to perk up. However, some 10 or 15 years after their move from Russia, the disease occurred again, affecting 5 of her 9 children. At first, it wasn't clear what this disease was, but eventually a very bright young man, a great-grandchild of Hannah herself, called Ben, had become a medical student and became fascinated with why it was that his mother and his grandparents and other relatives who he'd heard about had come down with this disease. And so he managed to acquire some brain tissue from one of the affected members of this generation here, and looked at it and realized that in fact she had what we now know to be Alzheimer's disease. Unfortunately, all of these people who came down with the disease died, and then there was a period of respite again until the 1950s, when once again this disease reappeared in the next generation, affecting 10 grandchildren. And this went on, and eventually the next generation came to the age of about 40. They too started to develop the disorder. At this time, Charles was becoming quite old and handed on the sort of family responsibility to Charles, who was also a pathologist. And Charles continued the tradition of keeping in touch with the family members that were scattered throughout the United States. He continued the tradition of archiving the stories of each of these people, and wherever possible, acquiring post-mortem tissue. And eventually, in 1980, some additional members now in this generation started to come down with the disease. And one of them went to see Dan Pollan at the University of Massachusetts in Worcester, who began to care for this patient. And by a serendipitous accident, Dan happened to hear a lecture that I gave about how one could use recombinant DNA technology to map genes causing diseases. And so he suggested that I come and talk to him about this family and see what could be done. And that started the next generation of investigations. But by this time, by the 1980s, there had now been 28 cases of Alzheimer's disease in the 5 generations that we knew about here. So in 1985, the hunt began. And with about 7 years of work, in 1992, we were able to report that the gene causing Hannah's disease was in fact mapping to this small region here of chromosome 14. At that time, however, we had no idea what the actual gene itself was. And so the next step was to try and clone the gene from that region of the chromosome. And as John Hardy told you, this is a very labor-intensive approach in which one has to clone each overlapping fragments of the chromosome and then interrogate each of these fragments for the genes that it contained, and then take each of those genes and sequence them and look for evidence of mutations in that gene that were present in individuals who had the disease. Most of these genes, including this one here, S182, which was the code name we gave it, had never been described by biology previously, and their function was not known. During this time, as the hunt progressed to find that actual gene, Charles himself became ill. He developed pancreatic cancer. And towards the middle of the spring of 1995, we began to get messages from Charles himself and from Dan Pollan that it would be nice if we hurried up a bit and found the actual gene. By the April of 1995, we actually had a fairly good idea that we had found the gene causing Hannah's illness. But we weren't entirely sure. And a few more phone calls were made to emphasize that Charles's mission in life had been to find this gene, to find the cause of the illness that affected his family. And so one day, late in April, when we were quite sure that it was correct, we called Charles and told him this good news. We weren't actually able to speak to Charles himself. He was very ill at that point. But we spoke to his wife, and the following morning she called and told us that he was at peace, that he saw a great sense of relief from knowing that his mission of finding the cause had probably been achieved. And indeed, he died a few weeks later, I think, relieved that what he had shown as an important goal for himself and his family had been achieved. We also found that Hannah's gene was also present, the exact same mutation, in another Jewish family living in America, but had come from a different region, from Odessa, meaning this is probably an ancestral mutation that had been shared amongst Jewish people of Russian origin. The gene in Hannah's family and in that other family encoded a protein that looks like this, with several transmembrane domains, a large loop domain here, and then two more transmembrane domains. Within a few weeks, we'd found that there was a homologous protein, which we called presenilin-2, which was mapped on chromosome 1, and it too was associated with mutations that caused Alzheimer's disease. And so this evidence together argued that these two genes likely had a function that related to how, in fact, Alzheimer's disease came about. Further work over the next several years elucidated that the presenilin proteins did not act alone, but they were in fact part of a high molecular weight protein complex composed of at least three other genes: nicastrin, APH1, PEN2, and presenilin. As we further characterized these protein complexes, it became apparent that there were these two very unusual aspartyl residues embedded within the transmembrane domain, which is typically hydrophobic, and this was quite noticeable. And indeed, later on it was discovered that these aspartyl residues are in fact the catalytic entities that cut substrate proteins in the membrane itself. We worked out how exactly this protein was assembled. It's assembled in a very stereotyped way. APH1 and nicastrin, which actually is a substrate binding protein, assemble first as a scaffolding complex, and then subsequently PEN2 and the presenilin moiety are added. The addition of this PEN2 is actually necessary for the next step, which is the autocatalytic cleavage of this loop domain here by these two aspartates to generate the active enzyme complex. It also became clear from the work of us and others that this complex mediates the cleavage of type 1 transmembrane proteins. Specifically, these proteins are usually cleaved first by another enzyme. In the case of APP, this other enzyme is called beta secretase, or BASE, and it cleaves a few residues off the surface of the membrane and releases this ectodomain here into the extracellular space. This membrane-bound stub then becomes the substrate for the presenilin complex itself. And what happens is that the cleavage occurs straight through the center of this transmembrane domain here, colored in red. And in so doing, it releases an intracellular fragment called the intracellular domain, and it also releases this transmembrane fragment, the N-terminal fragment of that residual stub. As we and others looked into this, it became clear that actually it was not a simple single cleavage, but in fact was a series of very complicated cleavages done by this enzyme. There was one that happened, the initial cleavage, which happens very close to the inner leaflet of the membrane. There are then another cleavage, the zeta cleavage, which generates two smaller fragments which are degraded. This fragment here, as I pointed out, is released into the intracellular space and typically is translocated to the nucleus, where it activates gene transcription. The final cleavage is the gamma site cleavage, which liberates the N-terminal fragment. And for most proteins, this N-terminal fragment is biologically inert and is usually degraded. However, for APP, as we shall see shortly, it is in fact the motor that generates amyloid and probably initiates the disease. Indeed, to reiterate this now, for the APP protein, there's this initial cleavage by base, which liberates this fragment. The subsequent sub of APP, APP-C100 fragment, as it's called, then undergoes cleavage by the presenilin-mediated endoproteolysis, liberating AICD and this amyloid component here, which then it tends to aggregate and to precipitate into form the amyloid plaques. I should point out, just going back to this previous thing, that there are two types of cleavage, and it seems that if the substrate protein is presented in one phase, that a series of cleavages occurs at residues 40, 48, 50, and 50 there. However, sometimes the substrate is swirled by half a rotation, and a minor set of cleavages then occur at residues 42, 49, or 50, and it turns out that the A-beta species that's generated on this minor cleavage product ending at residue 42 are much more prone to aggregate and are much more neurotoxic. As we looked at this, we and others showed that the effect of mutations in the presenilin protein was to promote this minor set of cleavages which generated A-beta 42, and in doing so, they increased the amount of A-beta 42, which is neurotoxic. And therefore, it seems that very subtle mutations in the presenilin protein alter this balance between A-beta 42 on the minor cut site and increase it, making, therefore, overproduction of this neurotoxic species of A-beta peptide. That's probably sufficient to explain how the presenilin mutations cause increased risk for Alzheimer's disease. However, it is clear from what John told you already that the presenilin complexes cleave many other transmembrane proteins, and some of those other membrane proteins are in fact themselves AD-related risk genes. For instance, SOL1 and TREM2, which you just heard about from John, are also cleavage product or substrates for the presenilin complex, and so it is possible that perturbations in the cleavage of these substrates also adds to the way in which people with PS1 mutations get Alzheimer's disease, and it may therefore be this complicated additive effect. It may be the explanation for why the forms of Alzheimer's disease associated with PS1 mutations in this complex are particularly aggressive. So now, in the most recent past, the questions have turned slightly now from how do these mutations cause the disease to now how does the complex work? How is it that these substrates gain access to the catalytic site? What are the mechanisms by which what are the mechanisms by which these different cleavage events occur? And how can we use this knowledge then to design selective inhibitors that could be used for the treatment of Alzheimer's disease? And in order to do this, we began to build a molecular picture, a structural picture of the actual complex itself. So we isolated these complexes and we spread them out on a carbon grid, and we visualized them using electron microscopy. And each one of these fuzzy, very small 9 nanometer sized little formations are in fact a single presenilin complex. And they fall on the grid in various different ways, some on their sides, some on their head, some on their bottom. And by taking these images, each one representing a different way in which the complex has fallen, and taking these images and then sort of reverse back projecting them onto onto a 3D image, one's able then to recapitulate what the complex would look like in 3 dimensions. And doing this, we've been able to show that in fact the complex is formed like a little snowman with a body here and a head here. There are some interesting features. There's a central cavity with a pore on one side and a cleft here. And the significance of this cleft is, as you'll see in a minute. So this is a rotating view, and you can see the cleft here, and this central cavity, and then this head domain here. And we've been able to take this and realize that this part, the body domain, is in fact part of contains these transmembrane domains, and that this head domain here contains the ectodomain of nicastrin, which is heavily glycosylated. And we can prove this by showing that antibodies to this ectodomain here bind to the head and make it bigger than the unlabeled complex here, proving then that this is the orientation of this complex. So the next question we asked was, how do some inhibitors work? And there are basically 3 or 4 different types of inhibitors. Some of them are rather boring. They simply bind to the active site, and that's not very interesting. Some of them, however, look like peptides, and they don't act at the active site. They act somewhere else. And we began to realize that we could use these peptides as tools to understand how the complex works. Specifically, we think that what these peptides are, or do, is that they get into the pathway in which a peptide substrate would normally take, and then they get stuck and they block the enzyme. And so we began to sort of work on this idea that if we could use these peptides, we may understand something about the gate mechanism that allows peptide substrates to get in and access the active site. So we began to work on this, and we made a couple of observations. First of all, it became clear that using selective detergent solubilization, that in fact the complex functioned as 2 half complexes, one here and one there. And we also realized that the substrates bind to the edges of both of these 2 complexes, or these 2 hemicomplexes, as seen here. So it binds at this interface between these 2. And that suggests then that this interface is a critical biological space during which important things happen. The next thing we realized was that the binding of compound E caused some obvious structural changes in the complex and probably caused it to be compacted. And we could show this by adding the complex by adding the enzyme to the complex, and then noting that if you had 2 fluorescent tags, that they would exchange energy between them more efficiently in the presence of this small compound, meaning that the compound had caused them to come together. And we could show again that this was the case. We also showed that if you added these compounds, that what happened was that if you increase the amount of compound, you decrease the amount of substrate that bound to the complex. In other words, what was happening was that the compound E was binding to the complex. It was causing the complex to shut, probably the lateral gate to shut, closing off the substrate binding site. And we could visualize this anatomically at the electron microscopic level, at 15 angstroms, by showing that in the presence of the compound E, this complex had changed its shape slightly. The lateral gate had closed, the pore had closed, and the head had rotated. And you can see it here, comparing these 2 things. Here's the lateral gate cleft, which is probably also where the substrate binds, and when in the presence of the compound, this cleft is closed, the head is rotated, and this pore has also closed off. So this began to tell us then a tentative model of how these inhibitors block a substrate access. Basically then, they bind, they close the lateral gate, that lateral cleft, and then the substrate comes along and goes boink, unable to bind, and reflects off the complex. So that's how these work. They work by altering the structure of the complex and closing off that binding site. But we also realized that there was an interaction between the substrate binding site inside the molecule and the substrate binding site on the surface of the molecule, and that specifically in the presence of a non-cleavable substrate, you could show that there was increased binding of this inhibitor complex. And so what we thought was happening is that in the presence of this substrate, it binds to the complex and it causes the complex to open up slightly. And so you can now begin to put these pieces of information together and get some idea of how this insidious little molecular machine works. So substrate binds at the surface, the interface between the two hemicomplexes here, and when it does so, it causes the complex to open up, essentially opening up a translocation pathway, and the substrate then moves forward into the complex. As it does so, it crosses the compound D binding site, which causes the substrate binding site to close behind it, sort of acting then as a turnstile or lateral gate. The substrate is now encapsulated inside the complex, and in that hydrophilic environment now, the reaction is able to occur. The substrate is cleaved, the pieces fall out, and the enzyme recocks and starts again. And it can go on doing this. So let me conclude by showing you that there are some obvious, rather dry technical advances that have come from this long thread of research that started with Hannah and ended today in Tel Aviv. It's clear, I think, from what I've showed you that the precedent complex is a dynamic, small machine whose structure is such that it forms this very atypical enzymatic activity of cleaving membrane proteins within the hydrophobic environment of the transmembrane domain, allowing them to cut key proteins in a way that's needed for signaling throughout life. And this is the basis of how bacteria and humans send signals within cells by cleaving proteins like Notch and liberating that intracellular domain which goes to the nucleus. It's also clear from what I've told you that subtle changes in the shape of that machine that are induced by mutations alter the way in which some of the substrates are cleaved. And in the case of the amyloid precursor protein, what it does is it favors the production of a slightly longer minor species which is prone to aggregate and is highly neurotoxic. And it initiates the processes that John has told you about that activate a cascade of events that leads to neurodegeneration and to the clinical phenotype of Alzheimer's disease. What I've also shown you is that we are beginning to understand at a structural, physical level how this complex works, and I think this is going to help us in the future understand how the disease comes about and bring hope, which I think is the gift of Hannah and her heirs. Is the hope that by understanding how this complex works, we can then design treatments that will specifically inhibit the production of neurotoxic A-beta peptides while not interfering with the normal function of this complex. I think, however, that there is a larger lesson to be learned from this story. I think the story tells us that simply horrendous things can happen to humans through disease, through religious persecution and genocide. And this is clearly what happened to Hannah and her heirs. However, I think you can tell from the story of Hannah and her heirs that by showing a massive amount of persistence, a huge amount of stoicism and bravery, a strength of character, a trust in their faith, a trust in their community, and a trust in their family, that they were eventually able to prevail against this. And I think that's true not just of Hannah and her heirs, but also for humanity. I think also Hannah's heirs who continued this fight against this, who continued to bring the problem of Alzheimer's disease and the problem of their family to medical attention, despite the fact that in the 30s and 50s, wherever they tried, they weren't able to get help. But they felt, people like Charles and Bell, felt that moral obligation, which I think is a human obligation, to fight against not only the disease, but also all of the other perils that that family went through. And I think it would be interesting to ask, if one could, back in time, what would Hannah and Shlomo have thought of the story that I've told you today? It's a story that started 170 years ago, a story that's crossed 18,000 kilometers of space, and has ended up being all about very subtle changes in a 90 nanometer a small molecular machine. I don't know what they said. I don't know what they would think. I couldn't speak to them. I can't speak Yiddish, nor even Hebrew, as you'll see in a minute. But I think you can tell from their actions that they would have probably said, remember the past and believe in the future. And that's subtly correct, not only about their particular story, but also about why memory is important, why understanding and treating Alzheimer's disease is important, and why science is important. Remembering the past, understanding what has gone on, and then believing in the future that we can change these things, I think is a wonderful message that comes from Hannah's story. So let me close by pointing out that this wasn't just my work, but was in fact the work of many others through several decades of time, funded by several different institutions. And finally, let me thank Professor Amos for his help in translating my slides. Thank you.
Thank you very much, Peter, for this wonderful talk and link to the history, the past, present, and future, with a lot of hope. And we are moving for our next speaker, which is Professor Danny Michaelson from the Department of Neurobiology, the Life Science Faculty, and the Sagol School of Neuroscience. Professor Michaelson is in combat of the Lebech Chair of Molecular Degeneration. His research focuses on the mechanism of action of molecular and environmental risk factor of Alzheimer's disease, with particular emphasis on sporadic Alzheimer's disease and on ApoE, which is the most prevalent genetic risk factor of the disease, as we heard. And Professor Michaelson is going to move us from genetic and to mechanism that Peter spoke to translational aspect. He will speak about translational Alzheimer's disease research at Tel Aviv University. Please.
Distinguished laureates, ladies and gentlemen, in looking at the laureates, especially John, if I may say so, and the way I'm dressed, I'm reminded of a story of two chimney cleaners who came down from cleaning the chimney. One was dirty, one was clean. And then what happened was they looked at each other and the clean one washed and the dirty one remained as he was. So I am dressed in the British European tradition of honoring the laureates, and the laureates are honoring us in being dressed the Israeli Mediterranean part, and I think this is a nice change. So that's just in a word of opening. How do I start this? No. I can't give the previous talk, it was very good, but I can't do it. Okay. Well, the word translation, we all know pretty much in the context of languages, to translate from one language to the other. But actually translation has a broader meaning, and it really means to switch from one set of values or system to another. And what I wanted to tell you today is our translation-related work. What do we mean by translation? Science in general, and Alzheimer-related science in particular, has a lot of the focus is on understanding how it goes, how it works, what is going on. And the word translation means to translate from understanding to doing. Now clearly, doing is related to understanding and understanding is related to doing, but the word doing means that you focus your work in trying to apply what has been studied and so elegantly presented in the previous talks, to try and apply it to something which can be translated to the patient bed to help the patients. So this is what translation research is all about. Now, this is who we are. There's an amazing group of 12, I think, or so independent research groups at Tel Aviv University who work on Alzheimer's. And if you feel my voice quivering, it's not just because I'm excited on this occasion, but I'm always afraid that I missed somebody. Okay, the list is so big, and I made all the efforts I can not to miss anybody, and I hope I didn't. So what you have here is many names, and you're clearly not supposed to read them right now and see who they are, but the general idea is to see that there's a big variety of things. And my duty here today as a representative of this team is, or this super team, is to present their work to you and to show you what they're doing. And I can give you the bottom line at the beginning, which is always good to know, which the bottom line is going to be that we are very heterogeneous. Because like many other people, we believe that there's not going to be a single bullet target treatment for Alzheimer's. This can be debated, but I think there's a lot of examples in other diseases that this is not going to work. And because of that, we think that one should have a heterogeneous approach and trying to hedge your bets so that you can take, whenever a breakthrough comes, you can jump in that direction and have the background available. So the name of people that I'm showing here, I've decided to break them down by discipline rather than by alphabetic order the way they were before. And the first discipline is people who will be working on the amyloid beta, or as we call them, the Baptists. There's the people who are devoting their work in the lab to A-beta as a focal point, A-beta and presilin as well, trying to see what they can do about this and help the patients. The second group whose names are mentioned over here, they are of another religion called the Taoists. They represent people who work on tau, which is a major pathological hallmark of the disease, and I will show you some of their work related to the possibility that we can interfere with the disease via the tau window. The third group, which I belong to, is agnostic, and we believe that maybe focus should be gone on ApoE4, which has been mentioned in the previous talks. So in addition to these three religions, which take place at the university, we also have people who say, don't focus on a molecule, focus on a process. And as you've heard in the previous talks, oh, sorry, did I show that? Yes. Don't focus on a molecule, focus on the process, and these are people who work on inflammation as a focal point in which you can do something about the disease. So what I want to do is share with you very briefly what those people are doing, so you get a feeling that we're working on a very heterogeneous mixture of complementary approaches. So the first person I want to mention is Becca Solomon, who is not here, but she's the grand dame of Alzheimer's research at our university. She was one of the original pioneers in taking the immuno approach to dealing with A-beta problem. So her work was to try and take what you've seen before, the amyloid, the A-beta deposits, and by introducing antibodies, which are the blue guys over here, to try and dismantle that. And just to show that she was really one of the early ones, her work dates back to 96, so it's almost 20 years ago where this thing was pioneered to start. But she's not the only one who works on A-beta. Antibodies are very good, but they're very big, and maybe we want to work also with small molecules. So there's a team at the university whose focus is small molecules that can block the activity of A-beta, and this is a very nice and elegant collaboration between a chemist, Udi Gazit, who is dealing with the issue of how A-beta comes together to form those filaments, and a biologist, Danny Sagol, who specializes in the animal models, the mice and the fly. And together what they're doing is testing the ability of small molecules to interfere with the formation of A-beta and to help the models. And one slide just shows the sort of results that they get. This is a fear response of a normal mouse. High means it's afraid. And when you take an Alzheimer mouse, it is less afraid because it does not remember what to be afraid of, and you can cure this thing with a drug. So this is what, I'll skip that. So this is what I wanted to say about the Taoists. I'm sorry, the Baptists. We have several groups focusing in this direction in different complementary approach. Now, how about the Taoists? The Taoists, I would first mention the work of Hagit Eldar Finkelman. She focuses on the enzymes that interfere with tau, which is GSK is one of the major, GSK3, one of the major enzymes, and their philosophy of their work is to try and develop small inhibitors that by interfering with this enzyme over here, shown over here, can prevent damage in a model and help the mice, and they have some interesting results showing that this is indeed the case. That, sorry, I'll go back. Ilana, your turn will come in a second. Okay, so she has those inhibitors that block the activity of the enzyme, and she can show using these inhibitors that if you have a behavioral test, that again, high value means that the animal remembers, and this is a model which does not remember, and when they give the treatment, it helps. So, in addition to Hagit's work, Ilana Gozes, who is here with us, has pioneered a lot of the work on tau, but her work really, when it started, did not have the philosophy, a tau philosophy. It was not a Taoist approach, but it was trying to look at cytotoxic molecules and trying to figure out mechanisms of cytotoxicity and protection, and she discovered two molecules which are shown over here, and when studying the mechanism, realized that these molecules, which were got into the game as a protecting agent are really working with the cytoskeleton, so they are tau related. So she belongs to the tau religion in that way of thinking. And her small molecules, what they can do, this is shown here in real patients actually, when this is a cognitive performance test, and you can see here high values is good memory and low values is less, and only the group that gets her drug can improve the memory, and this is in early Alzheimer patients. So this is what we can say about the Taoists. We turn now to the agnostics, and the next slide shows a famous agnostic guy called Alan Roses. Unfortunately, he is not a member of our faculty, but I think he deserves a slide to show his work, which has been very important and has been mentioned by other speakers. So what he has shown, that 60% of the patients, that's a very high number, carry this very specific form of ApoE called ApoE4, okay, as opposed to 20 or so of the normal patients. But not only that they have it, but it correlates with the clinical situation in the patients. So if we take a group of patients, many patients, different variety patients, that all of them at the age of 60 were still normal because the disease starts later on. And then we look at the patients as a function of the presence or absence of those ApoEs. You see that the green lines, these are patients that had no ApoE and the disease started at the age of 90, whereas if you're unlucky to have a double dose of ApoE4, the disease would start 20 years earlier. So this is a very prominent risk factor in the disease because of the high numbers and the high clinical effect. So some groups, in particular our group, is focused to work on this issue. But one thing to think about, when we say that a gene is bad, we have to make a Talmudic argument. Is it bad because it's bad, or it's bad because we lack a good thing? That's a very different story altogether. So look it over here. Here's our happy, good ApoE. Here's one possibility to be a bad ApoE. The bad ApoE has some token, some negative thing added to it, a headache, and that's why it's bad. But on the same logic, a bad ApoE could be an ApoE that lost something, okay? And this is not, as I called it Talmudic, but it's not Talmudic. It's very realistic in the sense that if this is the mechanism, you want to remove E4. If this is the mechanism, you want to compensate and bring something back. So there is some work at the university that addresses these issues, and this is myself, and we are addressing these issues by two complementary ways. We are trying to remove the bad guys with antibodies, and we're trying to compensate by doing a biochemical trick of lipidation to try and bring back some of the activity. So for example, just to show you a sort of result that one does, one has the mice which have the good or the bad ApoE, and you can subject them to one test which is very simple, which I want to explain, called object recognition. The mouse is first exposed to two similar worthless objects, two tins of Coca-Cola, okay? So it sniffs them, and then it very soon realizes there's nothing interesting in them. Then the next day, you take the same mouse, you put them again with two objects. One is the Coca-Cola tin, but the other one is, no, I wouldn't say Pepsi, but let's say a bottle of some juice, so, okay? So if the mouse remembers, it will go to the new one because it knows the old one is of no value, okay? So this is what you see over here. A good mouse with a good ApoE goes in preference to the new object, okay? Whereas our bad mouse has no preference, does not go neither to the new nor to the old, but if we treat them with those antibodies, we can impair the performance. So this is some more data which I will not belabor you. So when we do that, we can see that we can repair many of the features of the mice, both by behavior we can make it to reverse, and by doing some biochemical markers. Every talk has one slide that the purpose is not for the audience to understand, but just for the speaker to brag that they did a lot of work. So this is this slide here. So I'm not really attempting to stereotypically explain it, but you see many things are reversed, which means that the activity has been effective. So to summarize what I wanted to say, oh, sorry, I forgot a very dear friend who's sitting here. Because, you see, it just shows you how self-centered everybody is on their own work. Once I spoke about ApoE, I thought I finished, okay? So I apologize. And in addition to the three molecules, we have people who work on processes of such as inflammation, and Danny Frankel, who you've seen before introducing the speaker, is one of our leader speakers, and he's studying the role of inflammation, how when something activates that inflammatory system, it can affect the brain and induce loss of pathology from the brain. So this is work of Danny Frankel, and the last speaker I want to represent is Rubinstein, a dear friend. His work is actually, he did not start as an Alzheimer focus, but rather he was looking at microglia, trying to understand what is wrong in microglia that can affect its function. And this led him to the discovery that a certain molecule called CD38, that the presence or absence of this molecule is key in protecting Alzheimer models against Alzheimer pathology. So to put everything together, as I said, we have several approaches, and you know life has the problems of the rich and problems of the poor. So if you go to a talk when too much is going on there, you may at the end get lost and say, wow, I don't understand what the thing is. I want to have one question, one answer, one direction. But Alzheimer, we think, is not a simple disease. It's probably a syndrome. And there's many things lying behind the phenomena that the clinicians define as Alzheimer's. And because of that heterogeneity and because of what we've learned from other diseases such as cancer, we believe that there has to be a multitude of approaches at the same time so that maybe the one most successful from them will be effective under some conditions. Thank you very much.
Good afternoon. I was told to be short, so I'm going to read it, because there's a lot one can say about Professor Brenda Milner. The Dorothy J. Killam Professor of Neurology and Neurosurgery at the Montreal Neuro Institute and McGill University, Brenda Milner is one of the most important neuroscientists of the 20th century and the 21st century, and indeed, I would say, a legendary neuropsychologist. Her seminal discoveries in the study of human memory and the temporal lobes have radically changed the way we think about memory and have fostered and indeed revolutionized our understanding of how the brain works. Her research has also had a profound influence on the assessment, diagnosis, and treatment of individuals with brain disorders. And she made H.M. famous, although after 3 decades he never remembered her name. By merging neurology and psychology, or brain and behavior, Professor Milner pioneered a new scientific discipline, neuropsychology, later to become cognitive neuroscience, and we are all grateful to you for this. Dan David Prize joins a continuous and broad recognition of Brenda Milner's work of the last 6 decades. It is indeed a great honor to have you among us. Thank you for coming.
This is the good pointer. This is the good pointer. Yes. I don't really need this, actually. I need this. Well, good morning, ladies and gentlemen, colleagues, friends. I too would like to say thank you again, a big thank you to this organization and for this wonderful prize. Now we're shifting themes a little bit. Why is that slide on? How did that happen? I want to go back. Thank you. Yeah, that is right. I'm going back in time. I have been working, I'm still working at the Montreal Neurological Institute since 1950. And this gives you a look of what the neuro looked like. The first slide shows you what it was like today, but this shows you what it was like when I went to work there with the military annex. Oh God, I should leave that. I'm sorry about that. I must not use this. It's no good. I have to get back. This is
This is backward.
This is
forward.
This is backward.
No, forward.
And this is when this is forward. That's great. Excuse me, I'm sorry about that. So, this is the Neurological Institute, which was created actually by Wilder Penfield, a neurosurgeon, for the neurosurgical treatment of epilepsy. Now, of course, it's much more than that. It's covering the whole range of neurosciences. And that little building down there was the military annex, which when I went there, where most of the patients I worked with were housed, and doesn't exist anymore, and the building has grown and so on. It's a long time, since 1950, and that's when I started there. And the idea was, and here we're changing very much the focus from a progressive disease like Alzheimer to a sudden onset of amnesic syndrome as a consequence of as an unwished consequence of a brain operation. So Penfield was pioneering the surgical treatment of epilepsy, primarily but not exclusively temporal lobe epilepsy, but we will only think about the temporal lobes today. And my job for my PhD thesis actually was trying to find out the effects of removal. However, the critical thing here is that, of course, in the surgery for epilepsy, in there, you make your operation, you try to find out where the patient's seizures are coming from, and if it's a temporal lobe, is it the left side or the right? That's very critical. And then the operation is carried out on one side only. And so I had the possibility, back in 1950, of trying to find out some of the cognitive functions of the temporal lobes by studying a long series of patients, rather similar patients, operated on by Penfield. Now, I think you have to really have quite a lot of imagination to think of how little we had in the way of technology. We have, of course, no brain imaging, but we had EEG, which is critical for the diagnosis of an epileptic focus, and run by Dr. Jasper, a very gifted electrophysiologist, but with very, very primitive tools. So that the clues we got from the EEG were not terribly, always very helpful. We got the plain films of the skull and the shape of the ventricles, and that was about all we got about the brain until we saw inside it. So you have the patient seizures, you have me assessing the patient, beginning to see what might be helpful, and guided by what I knew about monkeys, actually. And then, seeing what deficits. The idea with surgery for epilepsy, by the way, is that since seizures can have an interfering effect in surrounding healthy cortex. If you remove that, you may have an improvement in overall IQ intelligence, and this is the case. It often happens. If you can control the seizures, you take away the interference with surrounding healthy tissue. But of course, you can't restore the capacities of the area you're removing, and so what you see after a surgery is a specific deficits related to the locus of the removal, with perhaps, if you're lucky, an overall gain in mental alertness and intelligence. That was the pattern. And of course, unilateral lesions. And as I say, no, now, forward or back. There, I've got it. So what you see there is my supervisor, Donald Hebb, talking to Wilder Penfield, actually in 1958, somewhat later, and you can see me somewhere up here. Yes, that's right, the young Brenda Milner at a conference. And it takes very little to make these slides go in strange directions. I'm not even touching it. No, but I'm not. What's happening? It's doing it. Look, I'm not touching anything.
I will do it from here. This one is done. You want this one?
Yes, I think I want to go back to Penfield and Hebb. There we are. And no, I can do it. Then that's forward, right? It's just a little. This is forward, just a little touch. I'm sorry about this. I'm not. This is silly. Okay, let's. No, oh, you know what does it? It's nothing to do, it's this. If I put my finger on here, it moves. I didn't know that. That's the whole problem. I shouldn't touch that. That's the entire problem. Okay, well, we're starting this talk again. What you have to realize is that you had no technological aids, and you had to make the best diagnosis you could of guess as to the laterality of the lesion. And if you had removals from the in the left hemisphere, which is Penfield's sketch, Rasmussen's sketch, of the left cerebral hemisphere. And for the purpose of today, I know there are probably lots of left-handers among you, but we will assume that the speech dominant hemisphere is the left hemisphere for now. And what you see here is a sketch based on many, many observations in different patients who are awake and being stimulated in the brain of the speech areas, the frontal lobe and in the posterior temporal region, parietal region. And these, of course, are mapped out in order to be spared, because it's much better to have epilepsy than to be aphasic. And so the typical removal would be anterior here in one hemisphere. And you get memory coming in rather early, because patients complain to you that they have some trouble with memory. It's not a serious trouble, but if a patient tells you he has trouble with memory, you don't say, oh, I'm sorry, I'm interested in perception, which I happened to be at the time. You start to study memory. And so beginning to study memory was because the patients complained of having subtle memory impairments, and we found them very clearly: impairment in verbal memory from removals on the left, and impairments in memory for faces and places and so on from removals on the right. But these subtle and rather mild deficits were a good exchange for the control of seizures. And then these removals also typically more and more involved the medial structures of the temporal lobe. When Penfield began, the neuro was founded in 1934, and when Penfield began, he was very cautious, and he just removed the neocortex, but spared the medial structures, and found that he couldn't cure the epilepsy that way in most patients. And so by the time I was working there, the removals also typically included the amygdala, part of the hippocampus, and parahippocampal gyrus on the medial aspect of the hemisphere. And all went well with these interesting, subtle laterality effects, until we saw a patient who came back because some of the patients who had been operated on by Penfield earlier had had just a very conservative removal, and they came back to have the temporal lobectomy completed. And and we had PB, this civil engineer from New Jersey, who had had just the lateral part of the left temporal lobe removed for his epilepsy, and he was still having seizures. And so he came back to have Penfield complete the standard temporal lobectomy, this which he did just by the removal of the medial structures of the left temporal lobe. Now, I had spent hours, I mean literally hours, testing PB on various cognitive and memory tasks before the surgery, a couple of days before the surgery. The day after the surgery, PB, who had a high IQ and a very good memory span and so on, said to Dr. Penfield, "What have you people done to my memory?" He couldn't remember what he'd had for breakfast, if his wife had been to see him that day. He had a continuous anterograde amnesia. But when I tested him, he didn't remember doing any tests with me before, but when I tested him with, and this is the standard finding, his IQ remained very high, and his immediate memory span, people often say, mistake the sort of amnesia we get after these temporal lobe removals as loss of short-term memory. It's not a loss of short-term memory. Short-term memory, as defined by William James long ago, is really a limited capacity span system. If I say 6291 to you, you're going to say them back to me. But if I give you a long list of numbers, you very rapidly break down. So one's immediate memory span is limited capacity, but if you keep within that capacity, you can keep it going until you're distracted. And this is completely normal in the patient HM you've referred to, I'll be talking about, completely normal in PB. And also PB showed the IQ was unaffected by the surgery and was high. He was capable of calculating, good spatial skills, all the rest of it. It's just that as soon as his attention was distracted, he couldn't remember anything more. And this is a very distressing happening for a surgeon in elective surgery. You know, if you're saving somebody's life by taking out a brain tumor or something, well, if they lose their memory, it's unfortunate, but they're still alive. On the other hand, if somebody has just had a few epilepsy seizures and is earning his living and then loses his memory in this elective surgery, this is not good. And also, it was puzzling. We were sort of startled by this. And a month or two later, we had another patient who had a left temporal lobectomy in one stage, and the same thing happened. He was a glove cutter, so fortunately he could go on working with his father, but he had the same loss of memory. And so Penfield and Jasper and I, this was back in 1953, we said, you know, what's happening? How can we explain these exceptions? And so we hypothesized that there must be an atrophic lesion in the hippocampal region of the opposite hemisphere, on the unoperated side. In other words, that the EEG had misled us, or something, the clinical signs had misled us, essentially that the operation had been on the wrong side, but that there was atrophy on the other side in the hippocampal region. Why did we say hippocampal? We said hippocampal because PB's operation had been in 2 stages, and the initial stage, which was just a neocortical removal, had not affected his memory. It was only the second operation, in which the medial structures were removed, that had produced this devastating and continuing memory. And so we had this hypothesis. I have to say that 12 years later, when this patient died, Dr. Penfield got the brain, and we had the hypothesis confirmed. But at the time, it was just speculation. So in 1953, we reported these 2 cases at the American Neurological meetings in Chicago. And just before that, the abstracts for the meetings were circulated to the members of the society. And a neurosurgeon in Hartford, Connecticut, Dr. William Scoville, read the abstract. And he phoned Dr. Penfield, and he said, I'm interested in what you and Dr. Milner have been describing, because I think it's what I have seen in patient in whom I did my operation. And I would like to invite Dr. Milner to come down to Hartford and study this patient and any other of my patients that may interest her. And Penfield said to me, would you like to do that? I said, you bet. And I started taking the night train down. That's how you went to Hartford in those days. And with whatever apparatus I could pick up from the psychology department at McGill to try investigating and studying. And that's how people very rarely ask why was Brenda Milner studying HM. You know, Brenda Milner was in Montreal, and when there were lots of psychologists down in the States, it was because of Penfield and I had reported these cases and made Scoville perhaps begin to take interest in the findings in the behavioral findings in his patients. And the rest of his history, a bit of history that we will talk about. So, now these arrows. Now what was Scoville's operation? Well, first of all, Scoville's operation was bilateral. We would never have done a bilateral symmetrical procedure. You can get along with one eye, one kidney, whatever, but you don't remove both. And so that's not usually done. But Scoville had developed this bilateral medial temporal procedure to work with schizophrenic patients. He'd done some work with frontal lobotomies and not like the results, and so that was what he was trying to do. And then this patient, HM, we can call him by his name now, Henry Molayson, he died a few years ago, at the age of 82. And he had a very, very bad epilepsy. Now, first of all, it wasn't temporal lobe epilepsy. He had very, very unlocalized, unclear EEG patterns. He had major seizures many, many times. He had a very, very severe epilepsy problem, which had interfered with his schooling and his work. And nobody knows, and I think even after his brain has been looked at, it's not at all clear why this guy developed such bad epilepsy. He had a fall from a bicycle at the age of 6, but it was a very minor thing, and started having seizures at 10. And so Scoville had been reading all about the operations from Montreal for epilepsy, and he wondered if doing his procedure, which he had, his procedure, he hadn't been studying these patients, but his procedure was essentially, here you see the medial structures of the temporal lobe, and he was going in like this, and sucking out for varying distances in both hemispheres, always the amygdala plus parts of the hippocampus and perihippocampal gyrus, going back varying distances, and not the lateral cortex. So it was a much more severe procedure than Penfield's because it was bilateral. On the other hand, it was less. It was not involving the lateral cortex. So that was his goal in these operations. So I went down in 1953 to meet H.M., who we're allowed to show now. This is him when he was about 27, when I first met him, or just after his surgery. And I worked with him for hours. He was whenever I was down there, and Sue Corkin, my former student, carried on, has written a book about him now, but carried on working with him over the years. And he was very pleasant and cooperative, and he was happy working with us, but he never got to know us. He never recognized you. He could be sitting in the waiting room, and I had just been waiting with him, and I could walk past him and he wouldn't know me. And it's very strange, actually, when he died a couple of years ago, we felt, I think all of us who had known him and worked with him, felt that we had lost a friend. And it gives you a strange idea about, I always think of friendship as a reciprocal relationship, but yet we all felt we lost a friend, and yet H.M. didn't know us. He wouldn't recognize us even. So it is a very strange experience. But he was very eager to help science and work with them. Now, the very first time that I met him, I was still sort of exploring, trying to find out, what one should be looking for in these patients. And so I thought, well, I'll give him, his IQ had gone up after the surgery. It didn't totally cure his epilepsy, but it did help. And his IQ went up, and he was above average IQ. And I asked him, I said to him, I'm going to give you a number. I remember this very clearly, and I'm going back to 1955. I said, I'm going to give you a number, and I want you to remember it. I'm going to go out and come back a little later, and you tell me the number. So I said, I want you to remember the number 584. And I went out, and I had a cup of coffee with Dr. Scoville's secretary, and I came back 20 minutes later, and I said, what was the number? And he said, 584. You see, I was very, very naive in those days, and I thought, well, I was surprised, you see. I thought, I said, well, that's very good. You do it. He said, no, he said, it's nothing. I mean, 5, 8, and 4 add up to 17. You divide by 2, and then you get 8 and 9. Remember 8, and then you divide 9, and you have 5 and 4. 5, 8, 4, he said, it's simple. And so then I said, do you remember my name and who I am? No, I'm sorry, the trouble is my memory. And so, and of course, a few minutes later, when we were working on something else, he had no memory of ever having been given a number to retain or anything. This is the essence of the disorder. So the question became, really to analyze what this patient could do and what he couldn't do and what the heart of it was. Now, in those days, people thought of memory as something that was represented equally all over the cerebral cortex. They didn't think of any sort of localized systems that might be important. Neurologists did, because they saw, hypothalamic lesions associated with amnesia. But psychologists did not. They were very, very influenced by Lashley's writing, and Lashley had published on equipotentiality and mass action, that the important thing was how much functioning healthy brain you had in a rat if you were trying to learn a maze, rather than the particular locus. So when I started publishing about and talking about patients having these very permanent and striking memory impairments in the context of good intelligence. See, this is what is different from Alzheimer's disease that we have been talking about. In Alzheimer's disease, the memory impairment is an early and conspicuous feature, but then, of course, a lot more is wrong with Alzheimer's patients than this system, and it gradually, of course, becomes much more of a general intellectual decline. Here you have a sudden effect in a young person of a surgical lesion that leaves you with this devastating effect on memory, and yet leaves the intelligence intact and so on. And you have the nerve to say that these structures are important when everybody knew that memory was a distributed function. So this was not readily accepted, and one had to look to try to find an animal model in order before anybody would really believe this. And so the search for some effect, something in a monkey. And there were many, many tasks that monkeys and rats with these lesions were able to learn. And so, but that's because they were learning them probably in different ways from the way in which a human being would learn it. But there was a breakthrough, and it took 17 years, I think, before all this was really accepted by Mort Mishkin and his group at NIH, and by a simple delayed matching task. If I were to show HM, I'd say, what is that? And he'd say, it's a blue bucket. And I'd say, well, I want you to remember that. And then during this interval of a minute or so, and then I show you these two objects, and I say, which is the object? Which is the new object? Well, HM would do that perfectly, and he'd do that perfectly because he'd have been saying blue bucket to himself. And this was true in any of he was very motivated to remember, and the lips would be moving, and you'd hear blue bucket. And then, of course, he wouldn't make any mistakes. But if you were a monkey, you couldn't say blue bucket. You couldn't verbalize. You'd have to keep some sort of visual representation alive sufficiently to guide you after a delay. And with this task, which is actually called delayed non-matching to sample, the monkeys failed miserably. And this was clear evidence that then the scientific world began to believe that really lesions in this area had an effect on memory. And so, and then a group in Massachusetts, in Boston, developed a rather elegant way of looking at this actually in HM by ellipse matching. You can show HM or show a patient an particular axis ratio. Then you can have a delay variable, and then you can say, pick out the ellipse that I just showed you. And if you have, this is not easy to verbalize, not easy to look at the ellipse, whereas he could look at the blue bucket and say, blue bucket, blue bucket, blue bucket. He can't look at a particular ellipse ratio and then a minute or so later pick the correct one out. And he failed miserably. After about 30 seconds, there was no relationship between the model that was given and the choice of the patient after the interval. So this was very much a human validation or a demonstration that we're indeed seeing the same phenomenon in monkeys and in people, and this became accepted that these structures were critical. I don't know. This is not even moving. This side is not moving. I'm not doing the wrong thing. It's just not. You see, it's not.
21st century technology is not working.
Well, I'm getting paranoid about this, these slides.
The computer.
Now it works. So we get to the situation in which people do now accept that damage, clean sort of surgical damage to bilateral, to the medial structures of the temporal lobe, produce this persistent amnesic syndrome in the context of well-maintained normal intelligence, normal behavior in other ways. Of course, a terrible handicap. But then you cannot go around and claim the universal negative. I couldn't go around and saying HM can't learn anything, because the next question is, what have you tried to teach him? And so that was when the challenge became that I had to find what he could learn and what he could not learn. And so that was the business of picking up tasks from psychology department and going down by train with them and spending 2 or 3 days working with HM trying to train him. And I took complicated mazes down on the assumption that he wouldn't be able to learn them, and he didn't learn them over 3 days. But I had a breakthrough with a sensory motor task, mirror drawing, which I took. In this task, the subject is given a sheet of paper, 8 by 10, with this double contoured star on the paper, and point S is the starting point, and he is told to trace a line keeping within the narrow borders of the star, but only seeing the star and his hand as reflected in a mirror. And if any of you have tried mirror drawing, it's really quite difficult and frustrating at the beginning, especially when you, at these points, you do this kind of thing, and you get very cross with yourself. But then over a few trials, the performance improves, and you get a very nice learning curve. And here you see what we found with HM over 3 days. This is his errors coming down. These are just interpolated trials with the other hand. And then the next day, he didn't, of course, he'd no idea he'd done it, right? He didn't know me. He didn't know anything. He was very ready to do tests, but he didn't know us. And yet you see how much he had learned. And the third day he has this beautiful performance. And at the end, on the last trial, he was a tall man, and he pulled himself up and he looked down and he said, "It's funny. I thought this was going to be difficult, but it looks as though I've done it pretty well." And so you realize that this was learning without any memory of him having done this procedure before at all. But the learning mechanism was continuing. So on the basis of this and similar tasks, we said, well, there are certain kinds of learning, that memory is not a general process. Learning is not a general process. There are different memory systems in the brain, and maybe motor learning is really different. You know, it does follow somewhat different rules. You have to practice a lot when you're young. It follows different rules of how you learn, and maybe it's a different system. And so Sue Corkin at first, and then other people tried many other motor tasks, and because you can't just generalize from mirror drawing, and found it is indeed true that motor learning, a lot of these tasks depend very much on cerebellar mechanisms and so on, but they are mediated by other systems in the brain and the medial temporal lobe. And then, shortly thereafter, my colleagues in England, Larry Weiskrantz and Elizabeth Warrington at the National Hospital, Queen Square, started using perceptual tasks, those puzzle things where if you're shown a puzzle picture of drawing, what is this? And we've all sort of scratched our heads over puzzle pictures, and then suddenly the light dawns and you get a bit more information in the contour, you're given a bit more, the picture becomes a little less puzzling, and you say, aha, that's a dog, And then after that, even if you're shown just the tiniest bit of that outline, you know immediately that it's a dog. And this is something which seems to depend not on the hippocampus, but on cortical mechanisms, posterior visual mechanisms in this case, and the cerebral cortex. So Warrington and Weiskrantz showed that with some patients, amnesic patients in England. and with herpes encephalitis. It became suddenly very fashionable. Everybody wanted to study these temporal lobe amnesic phenomena, and post-herpes encephalitis is an opportunity. And so in England, they were studying this intensively. And this, published in Brain some years ago, was the fact that these amnesic patients in England were able to show the benefit of having solved a puzzle picture, so that the next time they were shown a fragment of the contour, they needed less information. It was stable. And this has been, I replicated this in HM. It wasn't my idea, but I replicated it. And Sue Corkin has followed HM with these puzzles, and it was showing that this is very, very stable, and showing even months afterwards that the patient can recognize, know what he's seeing, although he's no idea that he's ever done this test before. Whereas, so it's quite impressive. So this kind of, and we think that these are neocortical mechanisms of learning and memory, and quite different from the motor skills, which are all quite different from this memory, which is what what we mean by memory. I don't know how many of you saw the film Memento, for example, which gives you a very good picture of what this amnesic state is like. And it's what gives the continuity to us in our lives, and we say it's our memories, but you see there are many, many other memory systems in the brain doing rather different jobs, and they are not affected by lesions of the task. Where one is going now with new techniques, I don't know the answer to this question, but we have now very fancy imaging methods. We have really a much better way of looking at the brain. We can ask new questions. With my two young colleagues who are with me today, we're trying to explore how the two hemispheres of the brain work together in memory processes. I had the good fortune to study Roger Sperry's patients with split brain patients at Caltech many years ago. And whereas Roger, quite rightly, and he got the Nobel Prize for this, very brilliant work, showed in these patients, whereas again epilepsy, we owe epileptic patients a great debt, because Sperry's patients had epilepsy and they were subjected to a different treatment, which was the section of the corpus callosum, so that the two hemispheres, each hemisphere was sort of functioning independently of the other. And he used this with very clever techniques to bring out the contrasting specializations of the two sides of the brain. And this was wonderful. But and I did some work there and published it on tactual memory with these patients. But what impressed me was that I would spend a few days at Caltech testing these patients and showing, and that I was always interested in what the right hemisphere could do. I'm a very verbal person, and I'm very impressed by good right hemispheres. And so I wanted to show that the right hemisphere, without words, could have a better memory of certain things, geometric forms and so on, than the left with its words. And these group of patients of Sperry's were perfect for that. And so with a colleague, Lachlan Taylor, we showed that this very clearly, we published it, we spent a long time working, go down to Caltech, spend a week working with this group of patients, and we had a nice study to publish, but of course you have to get some controls for these patients, so we came back to Montreal with these tasks, and this is a task on which the right hemisphere was doing better than the left. It was a tactile form matching where the subject puts his hand under a cloth and palpates a nonsense shape, and then after a few minutes goes back and finds it among a group of others. That's a tactile matching, delayed tactile matching. We saw delayed visual matching, and with the other hand. And we saw that the left hemisphere, that is to say the right hemisphere, the left hand, was performing better. So go back to Montreal. The patients had quite a lot of trouble with either hemisphere, you see, with these tasks. We go back to Montreal, and I get a few matched for IQ, not very high IQ, patients in Montreal who were about to undergo surgery for epilepsy. And I bring them down to do the tasks that we had just done with the split brain patients. And they say, But Dr. Milner, we know, Dr. Rasmussen says we have to do neuropsychology, we know we have to do these tests, we understand why we have to do tests, but these, they are so easy, they're so disgustingly easy. I mean, that we're wasting our time, we'd rather go back and read a book or watch television, and they were quite cross. And so I thought, well, it's obvious that, the corpus callosum is a pretty important structure, that if it's split, you can contrast the role of the left and right hemisphere in memory very beautifully. But then the fact is that neither hemisphere by itself is really, at some point you need help, even on a verbal task, there's some little imagery, little something. This is my hypothesis anyway, and it's actually what we're working on now. The two young ladies who are here with me today and we are working on hemispheric interaction. We're trying to test people it's just the beginning. They've done just a little pilot group with that, I think. They're just looking at the data, but putting patients in the functional MR with memory tasks, and which involve really both verbal and visual coding to do well. And so and we're interested in looking at individual differences in hemispheric interaction with imaging. But this is work I'm doing funded by the Boltzmann Foundation. They sort of asked me, what do you want to do? And I said, I want to look at hemispheric interaction. And so that's what next for us. But what next for the study of memory in general? HM has, as you know, after he died, he was looked at by Henis, I think, a neuropathologist in California. And it's true that Scoville did remove more or less what he said. He was a good surgeon, and the removal didn't go back as far as he had said it was, but it involved both the amygdala, the hippocampus, the parahippocampal gyrus, going back quite far in both hemispheres. And that there seems to be no doubt. There does not seem to be any evidence in HM's brain that he had some other very bad pathology that we did not know. We don't know why he had seizures. We still don't know why he had seizures. So this area will no doubt be pursued further. But for me, my present hobby, the thing that keeps me going and working still, is this question of how the two hemispheres work together in memory processing. And I think that it's very important, this interaction. And I think that different individuals use these strategies more than others. And so these are the things we want to explore. So thank you.
Thank you very much, Danny. And as Danny said, in order to understand Alzheimer, you have to tackle it from many directions, and this is exactly what we are trying to do in the Sagol School of Neuroscience, trying to understand how the brain functions from many directions. And we have the researchers coming from biology, from medical science, from engineering, from computer science, from linguistics, and from psychology, and this will help us to understand how the brain functions. And with that, I'm going to close the first session. I would like to invite you for a coffee, and we'll meet here at 11:30 again. Thank you. Thank you, Professor Miller, for this wonderful talk, taking us back in history, telling us some new stuff that you didn't know about HM. And we are really happy that you're here with us today. And we are continuing and going from memory loss to memory erasure. And the next speaker is going to be Dr. Segev Barak. Dr. Segev Barak is a young scientist and new recruitment of the School of Psychological Science and the Sagol School of Neuroscience. His research focuses on the neurobiological and behavioral mechanisms of alcohol and drug addiction. He's especially interested in the role of learning and memory processes in substance abuse and other psychopathologies. His work on memory processes in alcohol addiction was published in prestigious scientific journals and received several prizes. And he will speak today about, preventing relapse in alcoholism by memory erasure. Please.
Thank you. First of all, I feel lucky to be here, speaking after these legendary scientists here. So in a way, what I'm going to present is a bit kind of opposite to what the previous speakers presented, because they were working, they dedicated their lives to saving, rescuing memory, and I'm going to show how to destroy or how to erase the memory. And indeed, when we think of memory impairments, we usually, so we have the normal memory, we usually think of one pole of the impairment, which is when the memory is too weak or when we have memory deficits, and this is what we usually see in Alzheimer's and other dementias, and after neurosurgeries that make lesions or so on. The other pole of memory impairments is when the memory is too strong, or when what we can call abnormally persistent memories. That's memories that are unwanted and we can't get rid of them, and we see that in other pathologies such as post-traumatic stress disorder, when we can't get rid of traumatic memories and they come over and over again, and also in addiction, and I'm going to focus right now on addiction, and especially on alcohol addiction. And alcohol use disorders are among the most serious problematic health problems in the world. It was ranked recently second, I don't know how well you can see it, but second out of all the neurological and psychiatric disorders in terms of the amount of health loss due to the disease. The first one, by the way, is major depression. And it is also a major economic burden to society. The costs are enormous. And unfortunately, the pharmacotherapy for alcohol use disorders is not very effective right now. Now, when we take drugs or when we drink alcohol, even in the very first time, there are changes in the brain, and these changes in the brain are what cause us to act differently, perceive differently, feel differently, and so on. But in the first times, it's usually transient effects. There are changes, they go out, and we're done. But with the repeated chronic and escalating use of drugs and alcohol, usually of excessive amounts, there are long-term changes. Sometimes these changes are permanent in a wide spectrum of aspects, from behavior to brain circuits, neurotransmission, the biochemistry of the brain, and to the molecular level and gene expression. And these changes might cause, they mostly happen in the system of learning and memory, because that's the system that addiction share with the normal brain function, and they may cause memories that are related to alcohol or to the drugs to be abnormally persistent. And one of the worst problems in addiction is relapse, because most people that are addicts try to get abstinent at some point, but 60 to 80% of them will relapse within the first year, and in fact, about 50% of them will relapse within the first 3 months after trying to get abstinent. The main causes of relapse are some stressful event, but also what we call cue-induced craving, or in other words, memories that are related to alcohol, these abnormally persistent memories. So when a patient is trying to get abstinence, he might encounter some cues, memories that were previously associated with the reinforcing effects of alcohol, such as passing by his favorite bar, or the bartender, or people that he used to drink with, or simply smelling or tasting a little bit of alcohol. These may all cause very strong craving, evoke the persistent memory, and cause very strong craving, and therefore also lead to relapse. So the rationale behind what I'm trying to tell here is that if we can somehow get rid of these memories, we might be able to get rid of relapse, or at least attenuate it. And of course, the question is, how do we get rid of memories? So in the psychology clinic, there are two main behavioral methods to try to attenuate or get rid of memories: cue exposure therapy and aversion therapy. In acute exposure therapy, the patient is repeatedly exposed to the cue, to the stimulus that evokes this craving, such as the smell, repeatedly exposed to the smell of alcohol without being given alcohol itself. So eventually the craving decays. However, studies have shown that, in fact, this is not an unlearning process or erasure of memory process, but rather two memories are created. I mean, the previous memory is still there, and there is another memory now, so the cue evokes both memories, and we have a competition between them on the behavioral expression. And as long as the cue-nothing association wins, we have no relapse, but under some circumstances, the cue and previous reinforcement association wins, and then we have relapse. And the same goes for aversion therapy. In aversion therapy, the cue, the smell of alcohol or the pictures of the bars, are repeatedly paired with an aversive consequence, such as something that causes nausea or an electrical shock. However, still we have the previous memory of the cue and the reinforcing effects, and therefore we have a competition, and as in any competition, sometimes you win, but sometimes you lose, and there is relapse. So these two strategies have limited successful rates to prevent relapse, and we are looking for something that will be more successful by erasing or preventing or disrupting the very initial association between the cue and the reinforcing effects of alcohol. And of course the question is how. So for that I will briefly present a model that was introduced 35 years ago by Donald Lewis. So it starts with the new information that enters the system into the brain, into the short-term memory that Professor Milner was speaking of a few moments ago. So when we learn a new piece of information, it goes into the short-term memory or working memory storage in an active state, a few seconds, a few minutes sometimes, and then some of it decays and some of it is consolidated and transferred into a long-term memory storage where it's stable. And when it's stable, the memory is stable, people thought that it can stay there forever and for a lifetime it cannot be erased. Now, Lewis suggested that when memories are retrieved, the memories are actually reactivated, they become active again, and when they are active again, they are also changeable and level. And it actually makes sense because sometimes we need to update the memories. When you entered into this room this morning, you knew something about memories, now you're updating your knowledge about memory, and then after you're done with that, the memory is reconsolidated or restabilizes and moves back into its stable inactive state, and so on. When we retrieve it, it's becoming active and level. When we are done with that, there is a reconsolidation process and it's restabilizing again. Now, studies since then, and especially during the last decade or decade and a half, found that there is a window of opportunity, and it's important to say that it was found in a wide spectrum of mammals, from mice and rats to monkeys, and also in humans, that this window of opportunity that we call the reconsolidation window lasts 5 to 6 hours, during which we can manipulate the memory, and after that the memory is reconsolidated and is becoming inactive and stable. So if we are trying to manipulate the memory after that, after the 6 hours, we can't change it anymore because it's already stable. And this allows us to try to manipulate or interfere with the reconsolidation process, and if we succeed, we might by that be able to prevent the stabilization and the memory will be erased. So it seems that memories can be attenuated or erased, and one of the most robust findings, most of the studies were done in animals and most of the studies were done in fear memories. One of the most robust findings was that reconsolidation process depends on the synthesis of new proteins, the creation of new proteins in certain brain regions that are related to memories, among which the hippocampus and the amygdala that Professor Milner just mentioned. So we chose to focus on a protein called mTORC1, a mammalian target to rapamycin complex 1. And I'm not going to talk too much about the protein itself. I just want to mention that this protein controls a system that synthesizes or creates new proteins in the brain directly in the junctions between neurons, which are called synapses. So neurons communicate with each other through these synapses, and creation of proteins in these synapses are very important for the learning and memory processes. And indeed, the mTORC1 protein is implicated in learning and memory processes. And luckily, we have a drug called rapamycin that we can use, and this drug inhibits and prevents the activation of this protein, and therefore prevents all the cascade and can interfere with the learning and memory processes. So the first thing that we asked is whether when we retrieve memories that are associated with alcohol, when they are already well-consolidated memories, if we retrieve them, whether we see activation of this protein of mTORC1. And we did not do these experiments on humans, but rather in a rat model. And we first trained the animals to drink very high levels of alcohol in the home cage in a procedure called tubal choice. They had 1 bottle of water all the time, and every other day they also received a bottle of alcohol, of 20% solution of alcohol in drinking water. It's a little bit bitter and not too tasty, so they start with low level, and with time they escalate and drink very high levels of alcohol and reach very high levels of blood alcohol concentration. And after about 2 months we switch them, we put them in special chambers where they have to press a lever 3 times in order to an alcohol reward to be delivered into the chamber, and they can drink it. So then they have to work for the alcohol, and they press pretty well and drink high levels of alcohol there as well. After about a month, we subjected the animals for 10 days of abstinence. This models the abstinence period in humans, and then we reactivated the memories or retrieved the memories by placing the animals in the same chambers, the context where they drank the alcohol. This is the bar for them. And we also gave them a small drop of alcohol just to remind them of the smell and of alcohol by the smell and taste. So this is supposed to activate the memory, reactivate the memory, and to initiate the reconsolidation process. And we wanted to see if this protein is activated during this process. So we scanned the whole brain, and I'm not going to show all the data, but just one region, and this region is called the central amygdala. So the central amygdala is a region that is known to play a role in anxiety and in fear, but also in learning and memory. And what you can see here is a picture from a brain of animals that were not, of an animal that did not retrieve the alcohol-associated memories, whereas this memory retrieved the alcohol-associated memory. There's no alcohol on board, just the retrieval of the memory. And you can see here, I hope, the red staining. This is the central amygdala, where the mTORC1 protein is activated. So we knew this protein is activated, and the next question that we asked is whether if we prevent this activation, we can interfere with the reconsolidation process and make the memory disappear, not stabilize and disappear, and therefore we will have less cue-induced craving and less relapse. So we trained the animals once again in the same procedure, drinking for about 3 months, and then 10 days of abstinence and reactivation of the memory for 5 minutes. And after that, we injected them with this inhibitor of mTORC1, rapamycin, that prevents this activation of the protein. And then the day after, and 2 days after, and even 14 days after, we tested for relapse to alcohol seeking and to alcohol drinking. So I'm going to show just a little portion of the data. These bars show how much they press initially to get their alcohol, and then they underwent the abstinence, the reactivation of the memory, retrieval of the memory, and the injection of rapamycin. Now the blue bars here are the bars of the animals that receive, the control animals that received the placebo treatment, and you can see that they relapsed back to alcohol seeking and to alcohol drinking, and this was indeed paralleled by activation of the mTORC1 pathway, this protein, in the central amygdala. And when we prevented this activation with the injection of rapamycin, we showed that they did not seek for alcohol at all, and they considerably reduced the level of alcohol drinking afterwards. So it seems that the mTORC1 inhibition erases the alcohol-associated memories and can prevent relapse. So just before, okay, I'll summarize that and I will say just one word about rapamycin. So what we found, just to sum all this up, we found that the retrieval of the memory, this is already known, the retrieval of the memory initiates a reconsolidation process, a restabilization process of the memories, and that this process, during this process there is activation of this mTORC1 protein that controls the synthesis of new proteins in certain regions in the brain, and that if we prevent the activation of this protein of mTORC1, we can erase alcohol-associated memories and by that prevent relapse. Now just one word about rapamycin, or maybe a few more words. Rapamycin is an FDA-approved drug. It is used right now in humans to prevent the rejection of kidney transplant because it's an immunosuppressant through this mTOR inhibition mechanism. So the plus for that is that it can be used in humans already, and we know it's safe, but the minus is that we might have some side effects. So what we're looking right now, on one frontier we are looking, or not exactly we, but other people are looking for more specific drugs that will do the same thing to the memories without the side effects, and that's always something that we want to do. And in the lab we are looking for behavioral methods that we can do during the reconsolidation window, and by that try to erase the memories and prevent relapse. And with that, I would just like to thank my collaborators in UCSF and the funding agencies, and of course my lab members. Thank you.
Thank you. I would like to thank all the speakers of the morning session. And I would like to invite you all for lunch and to observe the poster of the students that are presented outside. Some of the posters, as I mentioned, are part of the Teva in Tel Aviv, or Teva and the Academy collaboration. Thank you. And we'll meet here at 2:15.